Morning. Hi. Hi. How are you guys? I know. Like, you got other shit to do. But we have to do this for an hour and a half. It's fine. Okay. So we're going to finish synapses and start uh, heart and vessels today. Okay. So let's talk about opioids. Did we start talking about them last time? A little bit? Okay. So remember, we did say that opioids were much bigger strings of amino acids. They're peptides. They're not proteins because they don't have the folding going on. But usually between 5 and 40 amino acids stuck together. So did we talk about different kinds of opioid receptors? No? I can't remember where we did. It was Halloween, lots of shit happened. I don't know. I've had too much candy and you have too. Okay. All right, so let's talk briefly about this. Okay, so the function of your endogenous opioids, right, the ones that you make, which include endorphins and enkephalins, they are your natural pain relievers, and also they sort of give you momentary, what you perceive as mental clarity, and inhibit your perception of your own limitations a bit. And evolutionarily, we talked about that being very valuable in a situation in which you just have to get out of the situation and worry about the pain and the risk later. The receptors that they deal with are opioid receptors. That's what we call them. And they're delta, kappa, mu. There's actually some more as well, but these are the ones we understand the best. And some of these are related pain relief, and some of them are related to dependence, because we talked last time about the fact that you don't just want to try to get away from the saber-toothed tiger once, even if it's damaging and probably beyond your capacity. You want to do it every time. So one of the things that happens with opioid release, endogenous opioids, and also opioid medications, is that they tap in and reinforce themselves with the dopamine pathway. And that becomes problematic when you're using them as medications, of course. And it's really the mu one. So just a couple of clinical connections, and then I'll tell you some of the sort of treatments. Morphine, heroin, codeine, OxyContin, Percocet, so many of them are either opioids that are derived from the opium poppy, yes? Or some of them are artificial, synthetic opioids, right? The synthetic opioids can be way stronger, like so incredibly strong. Fentanyl, for instance, is killing people dead left, right, and center. They're opioid medications, especially opioid medications that have any action at the mu1 receptor, tend to be really, really habit forming. But we've been using opium forever. So I just want to briefly, even though I don't usually ask you guys about it, I want to talk about like sort of some treatment, some rescue medications, and the kinds of things they do for opioid addiction. So the portion of the brain that seems to be really important in addiction is called the nucleus accumbens. It's in the middle of the brain. It's actually one of your cerebral nuclei. It's way in the middle of the brain, and it's involved in habit reinforcement. But just briefly, just because you guys will probably be interested in this, I don't usually ask you about this unless it's a bonus question, and then I might, because I can do anything I damn well please on bonus questions. Okay, so tons of opioids. That's not a complete list, of course, right? But opioid addiction and overdose treatments. So how do these things work? You guys have heard of methadone as an opioid addiction treatment, right? So what methadone is a long-lasting opioid that reduces opioid seeking. And the opioid seeking and those behaviors tend to be part of the problem with being an opioid addict. So it's got an analgesic effect, so the pain relief, very little pleasure associated with it, but it also keeps you from seeking out the opioid. So what it is a replacement, okay? So methadone is not getting you off of opioids as much as it is managing your opioid addiction so that you can have a life, okay? And it's federally regulated. It's really tightly controlled. Some people are addicted to methadone and actually get it illicitly. So it lasts for pretty much 24 hours, then you have to go back and get it again, right? And so there are methadone treatment centers all over the place. And sometimes that's as far as anybody's ever going to get, is that you are going to switch from whatever opioid you were on to methadone, and then you can usually keep your job, you can keep your life, you can probably not trash your finances. So that's methadone. Suboxone is actually a medication that they use to try to get you off of opioids, and it's an interesting one. It is a 4-to-1 ratio. Just think about this for a second based on what you've known. It's a partial opioid agonist, but it's a mu antagonist. So how does that work? What would it do? What would Suboxone do? Any idea? Go ahead. Block the dependence receptor, give you a little bit of pain relief, and also not have you have massive withdrawal symptoms. People can work themselves off of Suboxone. The other thing that Suboxone does is if you actually ingest opioids while you're on Suboxone, there's a medication associated with Suboxone that will make you yak your guts up, right? So that's sort of some negative reinforcement associated with it. And Suboxone can be taken long-term as well. Sometimes people can actually wean off of Suboxone. Even methadone, sometimes they can. It takes a while, right? So that's how Suboxone works. And then the rescue medication that you see signs up for all over the place, and this is an important one for you to know about, is naloxone. This is an opioid antagonist. It is a very, very high affinity opioid antagonist for the receptors. So when you take in naloxone, what it's going to do is really tightly bind to the receptors so that the opioids can't bind to the receptors. This is important in an overdose situation because remember, in an overdose situation, that opioids will shut down respiration. Yes? So what will this do? It will actually bind so that you are not overdosing, probably not going into respiratory arrest. This has no pain relief. It's not a long-term use medication. It's a rescue medication, right, to keep someone from overdosing. If you have boatloads of opioids in your system, you might have to use it twice, right, before the opioids actually clear out of the person's system. So if you have someone in your family or in your immediate circle who has an opioid addiction, it is not a terrible idea to go get this from your pharmacist, to actually have it around. One of the times, you guys have probably known humans long enough to know this, at least I have. When people tend to overdose on opioids, it's when they've gotten off of opioids and then get back on because they don't know about downregulation. So if, for instance, I was clean of opioids, I was an opioid addict and I was clean for 6 months, right? And then I have a day in which I go use opioids again. And I use the same amount that I used 6 months ago, but I have re-regulated my receptors, right? That's when people generally overdose. Does that make sense? And so this medication is actually really commonly used now. Interestingly, it actually got to be a political issue in some areas of the country, which is just shocking to me, in which people have decided, cops and paramedics have decided, especially cops in some areas of the country, that they're not going to carry it because because they think it's enabling addicts. No. Yeah, that was my thought as well. But anyway, that's how that one works. Whoops. Okay, so I don't usually ask you about any of those on the actual test, but I think that they're interesting for you to know, especially with our opioid addiction. And this state is really, really terrible for opioid addiction as well. So you will be dealing with opioid addicts. What? Oh, balls. I hate it when it does that. No, it's not. It's still on. Oh. Okay. Huh? Yeah, it just recorded oh balls. So that's good. I have people who are not you who actually follow my YouTube channel, And they're like, why do you talk like that? I'm like, why are you listening to my channel? This ain't even for you, right? And they're like, I've got like some people from really conservative countries, and they're like, I wish you wouldn't talk like that. I'm like, you know, you didn't pay for it, so don't complain. Beggars, choosers, go away. Okay, so let's see where we are. Okay, so that was opioids. Okay. All right, so the other ones, these are also neuropeptides, but I don't know very much about them. It's just that we're learning a little bit about them right now, and I'm going to put them in there so if you run into them later in your pharmacology courses. So CCK is a neuropeptide that regulates gallbladder contraction. They're learning a little bit more about it related to digestive abnormalities. And then this one is an interesting one, orexin, which is a neuropeptide. It's a hypothalamus neurotransmitter. So hypothalamus is not just histamine, and we've known about this one for a little longer. And it regulates sleep-wake cycles, and they are investigating it as a potential treatment for narcolepsy medication, which would be kind of interesting. And then there's substance P, and substance P is a neuropeptide that decreases GI motility. Opioids do as well. Opioids kind of stop you up as well. But this one is interesting because it may be a treatment for pain in the future, but it's involved not in pain perception, and that's what opioids did, right? Pain perception in the brain. It's involved in pain sensation, and it doesn't seem to tap into the dopamine pathway. So possible analgesic pathways for treatment in the future. And then, of course, we're going to get into the ones that we didn't cover as medications before. Go ahead.
Would that one, the substance B, would that potentially have a negative side effect where it could deaden you to actually helpful pain?
Potentially. We haven't actually researched it yet. They just figured out that what it did recently. So definitely some clinical trials are in order, right? Because some pain is good because it tells you not to do that again, right? Okay, so now let's talk about the endocannabinoids. So when I started teaching physiology, we didn't even talk about these as potential medications because they were just considered illicit drugs, right? Your THC, CBD, those kinds of things. In addition, interestingly, we didn't know about your own endogenous endocannabinoid system until 1991. But people have been using cannabis and cannabis derivatives for hundreds and hundreds of years, long before, of course, we knew about the receptors and we knew about the endogenous chemicals, okay? But conceptually, I hope it makes sense to you that you do not have receptors for any exogenous chemicals, yes? You have receptors for endogenous chemicals. And then the exogenous chemicals may act on a system that you already have. So you don't have opium receptors and you don't have THC receptors. You just don't, right? You have receptors for things that the natural world, right, might mimic, and then they would have some kind of physiological impact on your body. So let's talk about our endocannabinoids, the ones that you actually make. The one that you make, the big one, is called anandamide. Anandamide is a strange neurotransmitter because it's lipid soluble, which means it goes wherever the hell it wants, right? Okay. Ananda in Sanskrit means bliss. So this neurotransmitter was sort of named for the feeling of bliss that it causes. 1992, I said 1991. And for some of you guys, that seems like a really long time ago, but I promise that for some of us in here, it feels like yesterday, right? Or at least not too long ago. I was still a whippersnapper, but I was definitely alive and remember it. And some of you guys were born in the 2000s, and I don't even know how you're standing up. It doesn't seem like you should be able to, okay? This is an interesting one because in addition to the other functionalities and the reasons that people have used it, the pleasure functionality, anandamide is involved in CNS and PNS pain signaling. So let's do pain one more time. At least 3 possible ways to treat pain. Sensation, signaling, which is getting it in, and then perception. right? Opioids treat the perception, right? Substance P, at least theoretical, treats the sensation, and this treats the signaling, getting the signal in. Good? Okay. So so that's part of what it does in you. It's also, God dang it, what that blasted blast? What's happening here? Why are you doing that? Okay. It's also involved in working memory, short-term memory. It's involved in eating and sleeping patterns. It's involved in pleasure and food rewarding, but not via the dopamine pathway. As far as we have ever been able to find, this chemical and the exogenous chemicals that we use to mimic it do not tap into the dopamine pathway. Okay, so there is none of that chemical reinforcement with dopamine pathway. I'm not saying that it is not habit forming. I am saying that it is not chemically addictive by actually tapping into the dopamine pathway. So this is important to understand. Okay, so the receptors for your endocannabinoids are CB1 and CB2. They are cannabinoid receptors. And it's interesting that it took us so damn long to find them because they are among the most abundant receptors in the central nervous system. They are all over the brain. And if you have something that is both lipid soluble with ubiquitous receptors, then you can have a really fast response to it. Does that make sense? Okay, so of course the clinical connection that is worth discussing is anandamide, THC in marijuana, cannabis, right, is a weak anandamide agonist. That's what it does, okay? It's relatively weak, but it's also lipid soluble. You with me? Okay, so when you deliver it, however you deliver it, it has a really quick effect because of the lipid solubility and the abundant receptors. And it doesn't even look dramatically like, here's anandamide, yeah, and here's THC. It doesn't look dramatically like it, but it has some shape binding that actually fits into the receptors, right? So interestingly, it actually works really, really well. It doesn't bind massively, but it binds repetitively, right? So not super high affinity for the receptor, but binds repetitively to the receptors. And so, of course, you can talk about its use historically. So why do people use it historically? Well, historically, you've used it because it mimics this ananda, this bliss, right? But it also, of course, has pleasure and food rewarding involved in it. It does cause a little issue with your working memory, meaning you're going to forget what the hell you're doing half the time, right? You're going to have some amazing ideas sitting around on the couch, but then you're going to eat a pie and forget about them, okay? So they might have been the best idea you ever had, but you better tell somebody to write that crap down because it ain't going to last, okay? And also, they probably weren't that good. So eating and sleeping patterns, it can help with sleep in a lot of people. And then it's involved in pain signaling as well. So, of course, now, why are we looking at this as a potential medication? Well, because it is an analgesic that doesn't tap into the dopamine pathway. It also has some other effects that we don't understand yet. For instance, it decreases seizures in people who are predisposed to seizures. It can reduce the likelihood of manifestation of certain mental health disorders, like bipolar disorder in some people, not all people. But it is a pain management technique that does not tap into the dopamine pathway and tends to be relatively effective. And how about likelihood of, for instance, respiratory arrest or cardiac arrest by using cannabinoids? Damn near zero. Okay? So this is why we are finally, as a population, even in Utah, willing to have this conversation, right? And it took a while, right? When I started teaching here, I literally thought they would never even legalize medical cannabis. And we kind of legalized it, right? We kind of did, but then somebody said, you can't be trusted with those decisions, so I will do it for you. And I'm like, you can trust me before you can trust you, because I know more about this for a lot of reasons. But one of them is that I actually am a physiologist. So THC in marijuana is a weak anandamide agonist. CBD, which is, of course, legal here, has a little bit of the analgesic response, but less of the bliss response. It's also less effective as an analgesic, right? But it's not as fun. So, of course, we don't like fun things here in Utah, right? So we'll make that one legal because you can't enjoy yourself unless there's sugar involved, then you can go crazy, just bonkers. Okay, so go ahead.
With the food eroding, I know that they've used it in certain cases for eating disorders.
Yes.
I'm wondering though, kind of with certain anecdotal evidence, it
Okay.
seems that people who use it frequently develop abnormal eating patterns because it's like almost telling them when to eat. Is there something? Could there be something?
We haven't studied it enough. First of all, it wasn't Schedule 1, but there was no approved medical use. So now we have decades of research that we are going to need to do to figure out what that is used for. One of the things that it's really, really good for, by the way, long-term chronic pain often comes with decreased food responses, like you're just in too damn much pain to eat. And then that causes problems with healing, right? So those two are related. Like think about cancer, right? The pain that is associated with some cancers also reduces your appetite suppression. If you can hit with a double whammy of an analgesic that also increases your appetite, then your likelihood of long-term healing increases as well. Also, they do treat it with, anorexia. You can treat with cannabinoids if people are willing to do that. Go ahead.
This is anecdotal.
Okay.
But I had a patient who has chronic pain from a previous back injury, and he
Procured. Yes. Okay.
some of this and was on it like very small amounts over probably 3 months. We didn't see him, and he came back in and said that it was amazing and he wished it was legal here because he had such chronic problems with motility from his opiates that with this it helped with his pain and he was able to be more mobile and like move around and exercise and things, and also it deleted from his symptoms the constipation.
Oh yeah, yeah.
Chronic constipation.
Yeah. So it will be interesting, and it's going to be a treatment availability. Supposedly the dispensaries are supposed to be up by March. There's only going to be 12 for the entire state dispensaries, but I'm sure that there'll be one in Salt Lake, right? Because we're here. And apparently you can't go get it. They're only going to do deliveries at this point, but I don't know what the hell's going on. So they, anyway, so, and then just in case we're all feeling very, very superior, I would like you to know that part of the reason that you like chocolate is that there are weak anandamide agonists also in cocoa and chocolate. So literally everybody's a junkie for something. That's all I'm saying, man. So let's not be that uncool to each other. Okay. I forgot to tell you one other little thing. Sugar addiction, which doesn't seem like it would be a thing. It doesn't seem like a thing, right? Unless you're a type 1 diabetic, and then it's a thing. Yes? They treat sugar addiction because it taps into the mu pathways. They treat it with the same thing that they use to treat opioid addiction. So again, you're a junkie for something. Okay, so and then miscellaneous neurotransmitters. I'm not telling you that I know what these do. I'm telling you that they have discovered that they are neurotransmitters, and it's kind of interesting. Nitric oxide gas, not the giggle gas, but nitric, that's nitrous oxide, the giggle gas. And carbon monoxide, which will kill you dead as a hammer if you breathe it in, your brain makes it and releases it as a neurotransmitter. It just confuses the hell out of me. But it's not lipid soluble, so it's like doesn't go into your respiratory system, cause problems that way. So we'll figure something out about those later. Okay, so let's go into receptor functions here. So finding and naming neurotransmitters and receptors. Why are they called what they're called? Well, basically, it tells you something about the history of how they were discovered. Generally speaking, if you discovered a drug that messed with the receptor before you discovered the actual neurotransmitters and receptors, then everything's named for the drug, right? That makes sense? But if you discovered the chemical and then made drugs to interact with it, like the neurotransmitter, then everything's named for the neurotransmitter. So the only reason I tell you that is because there's no consistent naming convention because history isn't consistent. Yes? Good? So often the impact of a drug is known long before the receptor or the endogenous chemical messenger. So why are opioids called opioids? Because we knew about opium thousands of years before we knew about the endogenous chemicals, the neuropeptides, the enkephalins and endorphins. But like I mentioned before, you would have no inclination to repeatedly take the sap of that opium poppy and ingest it unless you had a system that it interacted with, yes? So the receptor must always have predated the drug, meaning that you should always be able to understand when I say that there are no opium receptors in the brain. There are receptors that bind to an endogenous chemical and also bind to opium, yes? And it's an interesting concept. I don't know why this tweaks my brain. I like the idea. Maybe the opium poppy and maybe cannabis are manipulating us into making sure that their population survives. Because guess what we do for them? Guess what we do? That's a good evolutionarily stable strategy, isn't it? Right? Just make sure that this entire population will make sure that you grow and they feed you and they clear land for you, right? And sometimes they'll give up every damn thing for you. So who's smarter, us or the opium poppy? Okay, they kill us and then we still grow them. Okay, so the drug, the opium derivative, can give rise to the receptor name, and that's why, for instance, you call opioid receptors, cannabinoid receptors, those kinds of things. Or if we knew enough before we started using drugs and we just used them because they felt good, then the endogenous chemical would have been named first, right? And then the drug would be named for that. So, for instance, GABA we found before we found benzodiazepines, which are GABAergic, right? Does that make sense? So they are anti-anxiety drugs, okay? So this is just how to put some logic to how the receptors in the drugs are named. Okay, so now we're going to go all the way back to chapter 5 because everybody hates this and you didn't want to do it at the beginning of the semester, so we'll do it really quickly. Okay, so here's the thing that I am about to cover for you. Signal transduction pathways. So what I'm trying to do is I am going to be an endogenous chemical, let's say a neurotransmitter, but it also could be a paracrine, autocrine, hormone, anything. And she's going to be the receptor, and you guys are going to be all the parts of the cell. And I'm going to try to make the cell do something, otherwise, why bother? Why bother talking to it, right? So what we are going to do is we're going to talk about the signal transduction pathways to get a cell response. Because can we agree that just because I bind the receptor, it doesn't actually make the cell do anything. There's something that has to happen inside the cell. Maybe protein synthesis, maybe opening an ion channel, right? Maybe endo or exocytosis, maybe programmed cell death, maybe any number of things. So there's a process that has to go on in the cell, right? And you call that the signal transduction pathway. Okay, and I think of it, my metaphor is always like playing telephone, right? I have to stay outside the cell if I am a water-soluble chemical messenger. She is stuck on the cell membrane. So how does she get somebody all the way at the back, like Grady, all the way at the back, to do something? If you're the nucleus of the cell and she has to stay there, what she's going to have to do is tell you something. You're going to have to tell JR something. You're going to have to tell Bree something. You're going to have to tell, and all the way back to him. Okay? Does that make sense? So these are signal transduction pathways. There are long pathways and there are short pathways. What do you guys think, just based on your knowledge? Which type of chemical messenger, water-soluble or lipid-soluble, is going to have the shortest game of telephone? Lipid-soluble. Because if I'm lipid-soluble, I skip all of you people. I find my receptor right there back by the nucleus, right? But if I'm water-soluble, I have to stay outside, and we're going to have to play a relatively long game of telephone. Good? So the first category that we're going to look at is signal transduction pathways initiated by intracellular receptors. Intracellular receptors, by definition, bind to lipid-soluble chemicals. That makes sense? Okay, so let's look at that one. So basically what I'm going to do, the primary mechanism of activating a cell response with a lipid-soluble chemical messenger is called gene activation. So look at this picture and then I have an animation for you as well. So I've got a lipophilic, lipid-soluble chemical messenger. Doesn't matter what it is, okay? It diffuses straight through the cell membrane because it can. It can find its receptor either in the cytoplasm or even all the way in the nucleus because it can go through the nuclear pores. You with me? And what it can do is it can directly activate protein synthesis by binding to a specific gene, okay, and then causing transcription, moving out, causing translation. Now I have a new protein. What is that new protein? I don't know. Depends on what the chemical messenger is and what the cell is. But here's an example. Testosterone, lipid soluble, correct? Yes? Testosterone, and this is a skeletal muscle cell. What could it do? It goes in, binds to an intracellular receptor, goes to the myosin gene, and causes protein synthesis of myosin, and then you have more myosin to make it more sarcomeres to build muscle mass. Does that make sense? So this is called gene activation. The chemical messenger goes straight through. The new ligand receptor complex binds to the DNA at the specific gene. For any of you guys who know that have anybody taken genetics? No, not yet? Okay. So there are binding sites at specific genes. So let me show you an animation that shows you how that works. So here's an intracellular receptor model. This one's also a hormone.
Aldosterone is a lipid-soluble hormone.
Okay, so this is aldosterone. It's lipid-soluble, right? Diffuses straight through, finds its receptor. That goes through the nuclear pore, the hormone receptor, binds to a specific region of the DNA, causes transcription, and then you have to send that back out the nuclear pore to a ribosome to cause translation. And then I have a new protein. What I do with the new protein depends on whether it was a protein that was structural or functional for the cell or one meant for exocytosis. But I definitely have potentially some new functionality to the cell. Are we okay? Okay, so that's called gene activation, and it's for blank soluble chemical messengers. Lipid soluble chemical messengers. Good. Everybody all right with that? Okay. And then there are way more water soluble chemical messengers than there are lipid soluble chemical messengers. So let's look at that. So the signal transduction pathways initiated by plasma membrane receptors. These are for water-soluble chemical messengers, and there are 1, 2, 3, 4 of those. And nobody likes them. We'll just get through it, okay? It's all right. We're going to be fine. You actually already know the first one. So one of the things that a water-soluble chemical messenger can do is bind to and open a channel that is also the receptor, right? So we call these, and we've been talking about them for a while, ionotropic receptors or ligand-gated ion channels. Yes? Okay. And of course, what's it going to do? It's going to open an ion channel, and then the ion is going to move across the cell membrane down its electrochemical gradient. Could hyperpolarize it, could depolarize it, could just be like calcium moving in to cause something to occur. But it's going to change the concentration of an ion in the intracellular fluid. And that is going to be the trigger for whatever the hell you want to happen. So what do you want to happen? Oh, I don't know, any number of things. Change in electrical properties of the cell, like induce exocytosis, use calcium as a second messenger. I don't know, any number of things. So the effect is that it changes ion concentration in the cell. Examples of these kind, nicotinic cholinergic receptors are excitatory sodium. They respond to acetylcholine. They are important in learning, attention, and memory. Those are the ones that degenerate in Alzheimer's disease. So these are called ionotropic receptors, and they are for blank soluble chemical messengers. Water soluble. Good. Okay. And then this next one is a receptor. Hold on. This is another showing you ionotropic receptor. They could do any damn thing they want once they get in there. Like calcium can do anything. Okay. So here's another one. Okay. This one, stick with me. It's not as hard as it looks. One end is a receptor. The other end is an enzyme. Yes? Okay. When something binds, the enzyme is activated. The enzyme being activated changes the activity of the cell by some mechanism, right? So you've got an enzyme that's doing something that it didn't do before. This category of receptors are called the tyrosine kinase receptors, and I'll describe them to you just a little bit. So a receptor that also functions as an enzyme, okay? Usually these are tyrosine kinase receptors. Why are they called that? because the way that they activate something is they have a protein with the tyrosine amino acid and they phosphorylate it, which is kinase, right? And then that protein now has some action in the cell. Good? Okay. So what did you do? You took an inactive protein, made it active, and now the cell does something different. Good? Okay. So this is an enzyme that is bound to the receptor. Good? It's actually part of the receptor, part of the same peptide chain. Okay. So this is how insulin works, by the way. Insulin binds and activates a tyrosine kinase receptor, which will ultimately cause the opening of a channel. A carrier. Okay, so, and then the next one is very, very similar to a tyrosine kinase receptor, and your textbook for some reason doesn't have a picture of it, but we can picture it together. Ready? So these do the same thing, only they are two separate proteins. So the enzyme is one string of amino acids down here, and the receptor is another, and they're stuck to each other. But they're not the same string of amino acids. With me? So they're just sort of adhered to one another. These are so ubiquitous that they are called JAK kinases, which they named for Janus kinase, as in Janus, is it Greek or Roman? The thing with two faces, is it Roman? A Roman? Two faces, but they started calling them JAK kinases, and somebody dubbed it just another kinase because there's so damn many of them that nobody is excited when you find one anymore. It's like, I found hay in a barn. Oh, good job, right? So what they do, same damn thing, only this protein is separate than this protein. They're called the JAK kinases, and what they do is the hormone is bound to the receptor. The bottom one's a kinase. It like kinases something, right? Phosphorylates something, and then activates activity in the cell. So hormones like growth hormone and prolactin, those bind to JAK kinase receptors. Okay, and then the last one, and this one's the most complicated. These are called G protein coupled receptors. Did anybody learn about these in 1610? Oh, good. So there's two people who are going to understand what the hell I'm talking about, and the rest of you are going to wait for me to shut up. But you know it hardly ever happens, right? Okay, so G protein coupled receptors. This is the most convoluted way to accomplish a simple thing that I could possibly imagine, because what is going on with these little bastards is this. Okay, ready? Okay. This is, for instance, an ion channel that I want to open. It doesn't have to be an ion channel. It could also be an enzyme that I want to activate. But what I'm trying to do is I'm trying to get this one to do something. But instead of going directly to it, right, I'm going to be passive aggressive. I'm going to ask 5 people to ask it to do something, okay? All right, so here's what's going to happen. I have the chemical messenger. It's water soluble, yes. It binds to the receptor. The receptor changes shape. I've got an animation for you in just a second. And what it's going to do is it's going to allow these G protein coupled receptors. The reason they're called G is because instead of using ATP, they use GTP as their energy source. Nobody cares, but that's why they're called G proteins, okay? And what they're going to do is they are going to slide through the phospholipid bilayer, the bottom part, and alpha, which they drew in the wrong place, it pisses me off, is going to slide into this one, smash into it, and that's how you're going to open it. So, in Texas, what do we call this? Going around your ass to get to your elbow. Like, why the hell couldn't you ask the damn thing to open on your own? Or it could do the same kind of thing, right? Only this one, I'm trying to turn an enzyme on, right? But I don't ask it directly. I use 3 friends to go over and ask it. It's like middle school crushes, right? You're like, I cannot talk to you. That is not allowed. And as a matter of fact, the whole time that we are dating, going out, talking to each other, we literally never talk to each other, ever. It's part of the rules, right? You can't talk to them after you're talking to them. So anyway, alpha smashes into this one to get it to do something. So let me show you an animation for that. I have a middle schooler. He's like, he doesn't understand. It's so delightful, this stage. He doesn't understand why people are dating. Did I tell you his quote about dating? He's like, I don't understand why people would date when they could play drums. And I'm like, bro, you have life figured out already at 13. He's like, they're spending all that time doing that. You could be playing drums. I'm like, yeah, play drums, please. Okay, you ready? So what this one's going to do, let me show you how it would work, is it's going to try to get this enzyme turned on, but it's going to do it the most convoluted way possible. So I'm going to have my water-soluble chemical messenger bind. I'm going to use GTP as the energy source to slide it through. It's going to smash into this little bugger, right? And then the thing's going to fall off. I'm going to hydrolyze the GTP to GDP. I'm going to use that to slide it all the way back over. I can't even fathom how this evolved. It confuses the living hell out of me. Like most of the time you can figure out, oh yeah, I can see how that would have evolved. This, I don't know what the hell's going on here. Right? And now it's ready to be used again. And here's the weird thing. There are so many of these. G-protein coupled receptors are so common. They're second after JAK kinases as far as like how common they are in the body. And once you figure it out, when you get brilliant and you get your PhD, send me an email and say, Mel, I figured it out. I'm getting a Nobel damn prize, but I'm emailing you first. Okay? And do. Okay. So those are G-protein coupled receptors. They are a huge group of receptors. They include muscarinic cholinergic receptors. Those are the ones that people figured out because people were dying of the mushroom poison. They include alpha adrenergic receptors that are found in the smooth muscle of blood vessels leading to the skin and the viscera. When norepinephrine or epinephrine responds to them, they shut down those blood vessels. They're found in beta adrenergic receptors on the heart. Those are inhibitory. And serotonin receptors are also G-protein coupled receptors. So for people like Rebecca who are going into pharmacy, these are going to be part of the thing that you have to master and understand, because there's so many of them that a lot of our pharmaceutical interventions are going to try to either have agonist or antagonist effects, right, at these G-protein coupled receptors. So they do end up being really important. Otherwise, I would never mention them because they're very hard to describe and everybody glazes over when I try. Okay, so to bring this back around to functionality, whether this is a hormone or a neurotransmitter or a paracrine agent, what happens at the end of signal transduction? Definitely something. Otherwise, why bother? But the something is cell specific, isn't it? So does testosterone do exactly the same thing at skeletal muscle as, for instance, it does on the cerebral cortex? No, right? But it does something. So kinds of things that it could do. It could, of course, activate transcription and translation and give you a new protein, right? That's one of the things it could do. Even water-soluble ones can do that, by the way. It's just that the water-soluble one, if I was going for the nucleus to start protein synthesis, I would have to stay here and you would play a long game of telephone to get there. It could activate or deactivate an enzyme, right, and change the activity. It could induce secretion of something you already had stored. It could induce cell division. For instance, growth hormone quite often does that. It could change the polarized nature of the cell membrane, depolarize, hyperpolarize. It could change the metabolic rate. And we could keep writing this list, right? Because it can do any damn thing. That's how you cells tell each other to do things. Do you think these are all of the categories of receptors that exist? Absolutely not. They are not all of the categories of receptors that exist, but they're the ones that I usually see students running into in the classes after this. So at least you've met them before. Cool? It's not everybody's favorite thing. As a matter of fact, everybody sucks about that part, even though I write super easy questions about it because they don't like it. And have you ever, I think I mentioned this to you guys before, everybody does worse on the last page of any set of notes because you flip, 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 and then you're like, done, right? And you never even look at the damn thing again. So look at the last page of notes. Okay, one last little thing, and this one's really easy, and then we'll move on to the heart. So I want to just talk a little bit about how nerve impulses, how your body, your mind, your sensory and motor systems decide to spend more action potentials on certain, let's say sensory, for example, certain sensory inputs than other sensory inputs. So let me give you an example. Right now, you are attending to my voice. You are using your cerebral cortex to sort of focus on my voice, right? And so it's sending relatively frequent action potentials because you're attending to it, but that's not the only sensation that you're taking in. You are also feeling all of your clothes on your body, but you're not attending to it, yes? It's a relatively constant stimulus. Is it really worth you sending very frequent, expensive action potentials for that? But do you send 0 action potentials? No. So what I want to talk about is how you do that, how you sort of decide what's worth more action potentials than other things. And it's not you that's doing it. It's mostly subconscious. Although in this situation right here, you are consciously deciding to do it, right? And you can consciously decide to like tune out too, right? If I piss you off, for instance, then tune out, right? Like when I piss you off, because it's probably happened once this semester. Okay, son of a... why is it even doing that? I don't even know. Okay, so basically, let's talk about how this occurs. Nerve impulses move down axons around 250 miles an hour. We already know that, okay? And they all start out the same way. All action potentials start out as a local or graded potential. Could be triggered by any number of things, chemicals, light, pain, sensation, whatever, any kind of thing. And you get a depolarized and graded potential. If it hits threshold, you get an action potential. All stuff that we know, okay? Once an action potential is produced, it is the same as every other action potential in that cell because there are no strong or weak action potentials, yes, okay, in a cell. Same intensity after threshold. But the magnitude of the initial stimulus is, of course, communicated to the CNS by frequency coding. We know that, right? So what's the difference between me yelling and ultimately what that is just frequency coding that's going into your brain, right? So to determine the volume that's going on. So it's frequency, not intensity. So what about this capacity for adaptation or accommodation? What about this capacity for your body to kind of ignore, but not really ignore, certain stimuli to attend to others? So for instance, have you ever driven through a super duper stinky town? Yeah. In Texas, it's all over. Because there's, some of them smell like tacos, and that smells like heaven. But also there are sawmill towns, and I don't know if you know what sawmill towns. They smell like butt foot. Is what they smell like. And you always wonder when you drive through one of those towns, like how the hell can people live here? Well, they can live here because of what's going on with this. Because after a while, you literally don't attend to the smell at all. And why? Well, what happens is that with adaptation or accommodation, the intensity of an action potential is constant, right? But the frequency will decrease once you have figured out that that particular stimulus is not falling into a few categories. First of all, new stimuli are always worth frequent action potentials because they have to be evaluated, right? So if you hear something new, so let's say you've got a caveman ook walking across the plains in Savannah, right, in the Savannah, and he is hearing the wind rushing through the grasses, right, and he's hearing birds tweeting, and he's hearing a babbling brook, okay? All of those stimuli are relatively constant and relatively consistent, right? So when you have a relatively constant or relatively consistent, like this, right, stimulus, then you can decrease the frequency of action potentials and not attend to it because it isn't changing. But if ook hears roar, that's a new stimulus, right? And it is going to go frequent action potentials until it is assessed. If it is assessed and you can eat it, it can eat you, or you can have sex with it, usually the action potentials will stay relatively high, even if you didn't want them to, right? So sort of subconsciously, those things that are threats, those things that are promising, right, stay relatively high. So what happens over time to a constant stimulus is that the receptors become less receptive to unchanging stimuli. And so you send less frequent action potentials to unchanging stimuli. And they don't have to be loud because all of us who are parents, if you think about yourself in the room studying and your kids are in the other room, right? And there's like kid noises going on. There's freaking Fortnite, somebody's swearing, whatever the hell your kids do. Mine are teenagers, so there's lots of swearing going on in the basement. I just pretend like I don't hear it. I don't really care, honestly. Lots of stuff going on. But let's say you've got toddlers and you're studying in one room and your toddlers are in the next room. They're close enough that you could pay attention to them. but you're not actually in the room with them. You, of course, are going to attend to the sound of the bookcase falling over, right? But does it have to be loud? Or what about silence? Right? So we are basically sort of wired up to send very frequent action potentials for new stimuli. We notice changes. Changes are worth the expense of an action potential. So we notice changes, but our sexes have evolved to be slightly different in our predisposition for adaptation and accommodation, which is interesting, okay? Historically speaking, males and females had rather different roles, right? So we tend to be communal. Humans tend to be communal organisms, right? And historically speaking, women were more likely to stay back with the group, right? They were going to stay back with the other women. They were going to stay back with the elderly people and the children, right? And so they had all of these different stimuli coming in at one time, right? So women, if you looked at the bell curve for women, they tend to be able to have multiple focal points at one time at the expense of single focus. So, for instance, I could sit with my husband at dinner. He could be talking to me. I could hear the entire conversation that's going on right next to me, especially if she's pissed, right? And he's like, why can you hear that? I'm like, why can you not hear it, right? So women tend to be better at multiple focus, but it is at the expense of single focus, right? They tend to be a little worse overall at single focus. Males, on the other hand, historically speaking, tended to, for instance, everybody else was back at home. They would go out, they would track the wildebeest, they would find the wildebeest, they would kill the wildebeest, they would drag that damn wildebeest back home, they would feed themselves first, let's be honest, then feed their family, then have sex, right? Go to sleep, do the same thing over again. So men, if you looked at the bell curve, tend to be better predisposed to single focus activities, right? And so you have to remember this if you are going to be in a relationship with someone who is the opposite sex of you, because their brain is not predisposed to the same things as you are, right? So, for instance, right, if you would like for your wife to pay attention because you need some focused bow chicka bow wow time, if there are 17 children in the house, your mother-in-law is coming over and the dishes haven't been done in 4 days, you are not going to get the single focus, right? Okay? However, typically speaking, if you would like to get a male single focus, you gotta be the damn wildebeest, man. You gotta hey, I'm the wildebeest. Especially if the wildebeest is super sexy, right? So be the wildebeest. So you basically have to provide something for the single focus. And you don't have to do any of this shit. You just have to realize that your predispositions are different. So for instance, my husband, who's a delightful human and a feminist, does not hear me when he is actually programming. And I will ask him to commit to something, and he completely commits to it and then tells me flat out that we didn't have the conversation because I was not the wildebeest, I was a gnat. And he was trying to get me to go away because he was dealing with his wildebeest, right? Does anybody else have that experience? He's like, I didn't say that. I'm like, I taped it. You told me that we were going to Fiji this summer. You said. Okay, so anyway, there is some difference. I don't know what's going on here. There is some difference with males and females in their capacities for adaptation and accommodation. Does that mean that you know what an individual's predisposition is? No. You're looking at a bell curve of predispositions that's in slightly different places for males and females. Does that make sense? Go ahead.
So when I'm studying, I put music on in the background.
Ooh, interesting. I can't listen to anything.
To tune out what's happening around me.
I turn on the TV. Yeah.
You turn on the TV?
Silence.
I lock the door, take away all distractible items, hide the remote so I can't turn it on. Especially if it's something that I hate doing. If I hate doing it, I have to remove all other things. Okay, so now we are going to get to the cardiovascular system. I will tell you that the cardiovascular system, I love it physiologically, but do not let yourself get behind because it's a bunch of little logical relationships that all build up to one huge concept map that I'll post for you guys. So I want you to, even those of you who are introverts, I want you to try to be brave enough to ask a question if you have it, or at least to write it down and solve it for yourself before you get back, or to email me about it if you literally would rather melt into a puddle on the floor than ask a question in class. And I understand, there's a few of you like that, but you have to solve the problem before you let it compound, because this is a bunch of little logical relationships. Okay, so first off, you have to know the A before the cardiovascular P will make sense. So if you do not remember the chambers of the heart, if you do not remember the valves, why they open, why they close, if you do not remember innervation to the heart, you need to go back and review that, because the physiology depends on the anatomy, especially with the cardiovascular system. So make sure that you do that before Thursday. Okay, so let's talk about the sort of physiological design, realizing that it wasn't designed, right, of the cardiovascular system. The cardiovascular system is really, it's actually relatively simple, because what you have is you have pressure, flow, and resistance. So what pressure does, is generate flow, because all fluids, blood included, will flow from high pressure to low pressure, right? That's why your sink works, yes? Okay, so that's pressure, which promotes flow, but then there is resistance, and resistance opposes flow. Resistance would be things like a gunked up tube, or a narrow tube, or high pressure that you're pushing against. So that's the concept of pressure, flow, and resistance. That makes sense? So it's really all just fancy plumbing. But the fancy plumbing serves one overriding function in the cardiovascular system. And this is really important for you to understand. The overriding function of the cardiovascular system is to ensure adequate blood flow to capillary beds, even if you trash the entire system in the process. Because let's bring it back to some physiology. If you can't deliver adequate blood flow to capillary beds, the tissues will not be able to do what? Cellular respiration, exactly. And if your cells can't do cellular respiration, then you're dead, right? Okay? So that is the reason why you will do things that are relatively counterintuitive, like this. Listen. So for instance, if you get arteriosclerosis and atherosclerosis because you've been eating too much Chick-fil-A and In-N-Out burgers, right? Because you're an American, and pew pew, freedom says you can. Go ahead. So you'll get arteriosclerosis and atherosclerosis, right? Which is going to reduce blood flow and increase your blood pressure. But since the ultimate goal is to ensure adequate blood flow to capillary beds, what will your heart do to compensate? Pump harder, which will increase your blood pressure. Yes? Does that make sense? So, and you can get just this system that increases, and it looks like it's a positive feedback because you're not looking at the right variable. This variable wins over all others, right? So you can get blood pressure high enough by that little process that you'll have a stroke or a heart attack, because the ultimate function of the cardiovascular system is to deliver adequate blood flow to capillary beds, even if you trash the entire system in the process. Because you were going to die anyway if you didn't deliver adequate blood flow to capillary beds, so you might as well invest in it. Does that make sense to you guys? Okay, so blood moves due to what we call bulk flow, produced by pressure. And of course, the pressure is created by the pumping of the heart, but there are two other factors in the pressure. Let's acknowledge them. So pressure is created by the pumping of the heart, specifically and mostly the pumping of the ventricles, yeah? Okay. But it can be augmented by vasoconstriction, decreased by vasodilation, right? By changing the diameter of the tubes. Anybody know one other thing that can really dramatically impact blood pressure? The volume. The volume in the fluid, right? So if it's plumbing, right, and you have very little fluid in the plumbing system, then the pressure is going to be low, right? If you increase the amount of fluid in the same system, then the pressure is going to be high. So basically what we are going to have is heart, vessels, kidneys that are going to be involved in this story. Does that make sense to you? Yes? because the kidneys determine your blood volume. And the kidneys do determine it by things like tonicity, and we'll talk about that as we get through. Okay, so as, God dang it. I don't even know why it's doing that. Okay, so basically, blood moves due to bulk flow produced by pressure. It's created by the pumping of the heart, of course, but it's augmented by a couple other things. And of course, we know what blood flow generally does. It goes heart, arteries, arterioles, capillaries, venules, veins, heart, with a couple of exceptions like portal systems, but that's mostly the pattern, right? Okay, so will you guys predict for me? As pressure increases, flow tends to do what? Increase, yes. Until pressure gets too high and you burst the system, yes? So there's a sweet spot for pressure, good, okay? And we'll cover that later. So flow is opposed by something called peripheral resistance, and let me sort of define this for you. And I don't want to tell you too much about it because I'm about to give you a think, pair, share about it as well. So peripheral resistance, so pressure is generated primarily by the heart, but resistance occurs primarily in the vessels. And what resistance is resistance is the opposition to flow. So what I want you to do in just a second, we're going to do a think, pair, share the first time. As resistance increases, flow tends to do what? Decrease, right? As resistance increases, flow tends to decrease. And resistance occurs primarily in the vessels. So this one might be easy for you, it might not. Let me go ahead and give you guys a shot at doing this one. Do it by yourself, and then we will do it together too. Do you know what atherosclerosis is? Gunking up of the vessels primarily with cholesterol deposits. 6 more people. One more. Okay. Okay, so let's go through that as we talk about resistance. So why is the ultimate function of the cardiovascular system to ensure adequate blood flow to capillary beds? Because, of course, gas exchange, nutrient exchange, waste exchange only happens at capillaries because they are the things that are thin enough and branched enough with enough surface area for this to occur, right? So the rest of it is really just fancy plumbing trying to get there. So every single cell in the body is within a few centimeters of a capillary. And so generally speaking, you guys know that in systemic circulation, capillaries are going to mostly be bringing nutrients and oxygen and picking up carbon dioxide and waste products, right? Okay, so once we get oxygen to the area, we don't have to worry about transport because it's lipid soluble, right? So it just moves down its concentration gradient. Nutrients move however they need to, right? They will move by facilitated diffusion or secondary active transport. If you needed to, you could move them by active transport. Waste products are going to move from the cell to the interstitial fluid back into the bloodstream. And then we also need to acknowledge that when you get to capillaries, you always do a little filtration, yes? Because the blood plasma is higher pressure than the interstitial fluid. And so as you go through a capillary bed, you are always going to lose some fluid volume, which is one of the many, many reasons that veins are going to be lower pressure than arteries, right? You always lose fluid. you will pull most of that fluid back up into the lymphatic system, and we'll talk about that later too. Okay, so let's get into the whole nitty-gritty of pressure, flow, and resistance. Okay, so the equation for blood flow is F equals delta P over R. F is flow, delta P over R. F is flow, P is pressure, and R is resistance. Okay, and of course, this is what we are trying to accomplish, yes? Okay, so what we are going to have to deal with is the flow of blood is going to be determined by two factors. The delta P is more important than the P for reasons that I hope will become obvious. So if I have a blood vessel, okay, this is a blood vessel, I don't care which one it is, and I have pressure at two different points. It's not the pressure that matters, it's the difference in pressure that matters, because look at this. If P1 was at 100 millimeters of mercury and P2 was 100 millimeters of mercury, do I have any flow? No, right? Because they're both the same pressure. But if P2 is 80 millimeters of mercury, then I know both the direction of the flow, and I could also predict a little bit about the speed of flow if I compared 80 to, for instance, 20. That would be a faster flow. Does that make sense to you? So intuitively, you know that it's the difference in pressure between 2 points. So really, it's not that pressure makes flow, it's the difference in pressure between 2 points. So why does, for instance, the blood flow from the left ventricle to the aorta to the brachial artery? It is because the pressure is highest here, lower here, causing unidirectional flow of blood through the system. Does that make sense to you? So that's what the delta P is. So blood flows from high pressure to low pressure, and let's revisit this concept, and then we'll look at a picture of it. Pressure is, of course, generated by cardiac muscle contraction. We know that, right? And will also be dramatically impacted by blood volume changes and changes in the size of the lumen of the vessels. Does that make sense? Okay. Pressure is measured in millimeters of mercury, whether you're talking about barometric pressure or blood pressure. Okay. And it is the difference in pressure between 2 points that determines both the direction and the velocity of flow. Is that logical to everybody? Right? Plumbing is relatively logical to most of us. It's like, I got this, okay? So the truth is, I'm not even touching it, little bastard. The truth is not that flow is proportional to pressure, but that flow is directly proportional to the difference in pressure between 2 points. Or not as pressure increases, flow increases, but as pressure difference increases, flow increases. And this is important for us to know when we start thinking about arteriosclerosis and atherosclerosis. So I want to look at this situation right here. Ready? Okay, so this is trying to show you the delta P in 3 different situations. So in this situation, I've got 100 millimeters of mercury of pressure over here and 60 over here, which gives me a delta P of 40. And if the resistance is 2, that gets me a flow of 20 mils per minute. Everybody okay with that? Okay. This one, I got a delta P of 0, so I don't even care what the resistance is because you ain't getting any flow. This person right here has been eating too damn much In-N-Out burger, because what happens is once that P2 increases, the P1 has to increase in order to still have a delta P. This is how hypertension occurs. Because, of course, if I get atherosclerosis, for instance, I gunk up my blood vessels, the P2 increases. What does my heart have to do to overcome that? It has to increase the P1, but notice the same delta P, the same flow at much higher pressure. Here's a question for you. Which of these is sustainable long term, A or C? A, yes. Because we know plumbing even, right? Super duper high pressure is harder on the system. Something's going to break, yes? Okay, so this situation right here, C, is what happens in order to maintain adequate blood flows to capillary beds when the pressure that you're pushing against creeps up. This is the process that leads to hypertension. Good? Okay, so All right, so flow is directly proportional to the difference in pressure between 2 points. The magnitude of the pressure in the system, if we're just talking about the systemic circuit, and assume that I'm talking about systemic unless I say pulmonary, the magnitude of the pressure in the system varies, and it has to, otherwise you wouldn't have unidirectional flow of blood. So without even knowing it, you can probably figure out that the pressure is highest in the left ventricle, lower in the arteries, still lower in the arterioles, lower than that in the capillaries, lower still in the venules, lowest in the veins, right? Super duper low in the superior and inferior vena cava, and it's about 0 millimeters of mercury in the right atrium. Does that make sense? And that promotes the unidirectional flow of blood. It allows you to predict which direction blood is going to flow. Is that okay with that? Okay, so now, so the magnitude varies throughout the systemic circulation. So let's look at what it does, both average pressure and then more data points. So if you look at average pressure throughout, for instance, the systemic circuit, the pulmonary circuit has a similar pattern, but it's way lower pressure. So what you see is the average pressure is highest in the arteries and then it falls, right? All the way to pretty much 0. The pulmonary circuit, question for you, see if you can verbalize it. The pulmonary circuit is way lower pressure. So my question to you is, why is it way lower pressure? Meaning, what anatomically is responsible for the lower pressure in the pulmonary circuit compared to the systemic circuit? Not why is it okay? It's okay, I'll answer for you, because the heart and lungs are right next to one another, right? But what anatomically is responsible for the difference in pressure between those two circuits? No, shorter distance is why it's okay. Anatomically, what's responsible for it? Yeah, the right ventricle has much less musculature, right, and generates less pressure than the left ventricle, right? Does that make sense? Okay. And the reason it's okay to have that look is because they're right next to one another, right? So the anatomical is responsible for this physiological difference. But it's not only lower as it moves through the system, but it's also most variable. If I take a few more data points and don't get an average, what I can see is the pressure is highly variable in the arteries, right? Right? You actually have pulse pressure in the arteries, okay? The highest pressure that you see in the arteries would be right after ventricular contraction. That would be systolic pressure, yeah? And the lowest pressure that you see in that artery would be right before the next ventricular contraction. That would be diastolic. That makes sense? So, do you generally measure blood pressure in a vein? No, it's not very variable. You could measure it, but you ain't going to get 2 readings, right? You're going to get 1 reading. In the arteries, you have highly variable pressure. In the arterioles, you're getting a little further away from the pump. It's not very variable. By the time you get to the other side of the capillary, you really have almost no variability, no pulse pressure that you can measure in the veins. Is that okay? So it's not only higher in the arteries, it's more variable in the arteries. Okay, so the blood pressure is, of course, as I mentioned, primarily impacted by volume, cardiac output, and resistance. So let's talk about resistance, okay? So resistance is really defined as how difficult it is for blood to flow between 2 points at a given delta P. So what I'm going to say is I want to hold this delta P constant, and I want you guys to think about what could increase or decrease the resistance between these 2 points. So let's go through it. You ready? So peripheral resistance, how difficult it is for blood to flow between 2 points at a constant pressure difference. So flow is inversely proportional to the resistance between 2 points. You know about directly and inversely proportional, right? That means that if the resistance goes up, the flow does what? Goes down. Exactly. Okay. Or as resistance increases, flow does what? Decreases. Exactly. So the primary consideration for resistance is friction. Yes? Okay. So what happens between these 2 points depends on how much friction there is between those 2 points. I don't even have to hold the distance of the points constant. So friction is the primary consideration in determining resistance. Most of the resistance, of course, is in the vessels, not the heart. The heart chambers are relatively wide open. The endocardium of the heart is usually pretty slippery. Okay, so what kinds of things can actually impact resistance, friction, and therefore resistance? The viscosity of the blood. Do you know what viscosity is? You talk about viscosity with motor oil, right? Viscosity is resistance to flow within the fluid. Two different fluids. So whole milk would be the normal kind of consistency or viscosity of blood, but it can get super duper thick like maple syrup. Okay? Which one is going to have more friction and resistance to flow? Maple syrup. So how could, for instance, that occur? Resistance to flow due to the friction. This changes with dehydration. So if you lose fluid, you don't lose the cells necessarily. It also changes with a hematocrit change. Hematocrit measures the amount of solids in your blood. For instance, if you had leukemia, you would have so many solids in your blood that it would be really, really viscous. Does that make sense? And it's relatively constant in healthy individuals. So your viscosity doesn't change dramatically over the course of a day or a week or anything. But you can get dehydrated. Okay, vessel length is the other one. So vessel length is a longer tube gives more friction. So if P1 and P2 are further separated, then it's going to give more friction. Think about that if you were trying to imagine blowing milk through a straw. If you are trying to blow milk through a relatively short straw, then you don't need to generate much pressure to get over that resistance, yes? But if you're trying to blow milk through a 4-foot straw, the same pressure will not be able to overcome that resistance, yes? So this matters, especially as you gain weight. As you gain weight, you make more vessels, yes? So let's talk about the correlation that is really, really consistent between weight gain and hypertension. So it's really a completely logical relationship, isn't it? If you gain 40 pounds, you have to have vessels to supply 40 more pounds of you. Okay, that's a lot of capillaries. In order to overcome the resistance with those vessels, quite often your pressure has to pump up. Does that make sense? Because it's F equals delta P over R, and your resistance went up with 40 pounds of weight gain. That also, of course, matters with women when they're pregnant. That's a temporary weight gain, but doesn't your blood pressure generally increase when you're pregnant? Because it has to, because you've got to feed like 20-ish more pounds of you, right? 20 to 40 more pounds of you. So that's the vessel length. It's generally constant in adults, except in instances of weight gain and weight loss. Go ahead, Jen.
I also noticed genetically with tall people have more circulation because really, honest, truly.
Yep.
My husband's, he's 6'10", his sisters are 6'4", and they all have circulation problems.
Yeah,
And
it's a little hard on your cardiovascular system to go that far and come that far back.
Genetically.
Yeah, tall people do. It's hard on your heart to be tall. So if you're going to be tall, you might want to be athletic.
You're supposed to have a shorter lifespan.
You do have a shorter lifespan, yes. But not individually, just as a group. You're fine. Okay, last little thing. We'll do the other thing, pair share, when I see you guys on Thursday. The other thing is just the vessel diameter. So think about this. If I squeeze this one, is there more resistance between P1 and P2, right? If I stretch it, there's less resistance in P1 and P2. So there is what we call the inherent vessel diameter, just meaning that bigger tubes have less resistance, right? So the aorta has less resistance than the brachial artery, less friction, less resistance. So that's the inherent vessel diameter. But then there is also vasoconstriction and vasodilation, which can temporarily change the resistance, right? And therefore change the flow. Is that okay? Okay, so we'll pick that up next time. Is your quiz due today? Okay, make sure you get it done. There's going to be another one coming. In addition, I'll probably post for you guys a big cardiovascular system concept map. If you hate concept maps, don't even look at it. But if you like concept maps, then it's a good way to study. We're going to do it again on Thursday. We're going to split that one up into two.
precise dose. That's all I got.
Oh, synaptic testicles, the ones that are actually
Still
still have
in
soul.
there?
Yeah, but you don't have to be free. It just has three chances to get the right one. I didn't do both of them if you want. It's fine. There's two. Neither one is correct.
No, it was the mouth. Yeah.
Well, that's what you should do.
Well, that's not